Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast is going to cover the cell cycle, and so I'm going to talk about what the cell cycle is and what it does, and really dig into the different phases of a cell cycle. There's two, which is going to be interphase and the mitotic phase, and each of these, of course, is broken down into different stages that I'll talk about. And then I'll talk about regulation of the cell cycle, which is important to understand. And then lastly, I'll talk about a few applications in terms of diseases that the cell cycle is involved in. And this is really just to kind of help you remember some of these concepts. If you can somehow tie these concepts into a disease that you already have some understanding of, then you're more likely to recall it on the MCAT. And this material is likely to show up in one of the four MCAT sections. That's going to be the biochem section. So with that said, I hope this podcast and the others are helpful as you study for the MCAT. So the cell cycle is a series of events that a cell undergoes that involves both growth and cell division that produces two new daughter cells. And as you'll come to see, this is a pretty carefully regulated process. And in terms of the type of cells I'm going to be talking about the eukaryotic cell cycle. So bacterial cells don't have a membrane-bound nucleus, so they do things a little bit differently. So just keep that in mind. In this podcast, I'm talking about eukaryotic cells. And as I was saying earlier, there are two main phases of the cell cycle. The first phase is known as interphase, which is the longer phase of the cell cycle. And then the second phase is known as the M phase or mitotic phase. And this is a little bit shorter of a phase. And interphase is further broken down into the G1 phase, the S phase, and the G2 phase. And I'm sure you're probably already familiar with the mitotic phase that's broken down into prophase, metaphase, anaphase, and telophase. And I'll go over those for those of you who don't really have a good grasp on what exactly goes on during mitosis. And I also wanted to put in perspective how much longer interphase is than the M phase. So interphase takes about 20 hours to complete, whereas the mitotic phase takes about 2 hours to complete. So overall, interphase is about 10 times longer than the mitotic phase. And if you do the math, overall, the cell cycle takes approximately 24 hours. Although I was reading that this is very cell dependent. So, if you're a cell in the liver, you might take more time than that or less time than that. If you're a cell in the lung, you might take a little more, a little less. So it's very variable how long the cell cycle is through different cells in the body, as well as a cell can also enter into what's known as the G0 phase. And I'll talk a little bit more about that in a second. But first, I want to go through and break down the different phases of the cell cycle. So as I said earlier, there's two main phases of the cell cycle. The first being interphase and the second being the mitotic phase. So the interphase breaks down into three distinct phases itself. That's the G1 or gap1 phase, the S phase, which stands for synthesis phase, and then the G2 or gap2 phase. And so what I want to do is I want to go through these steps sequentially in the order that they would happen. So let's just say that you're a cell and you're starting in G1 phase or gap1 phase. So the gap1 phase is actually called the gap1 phase because microscopically speaking, you can't really tell that there's anything actually going on. It looks as if the cell's just kind of resting, hanging out in this part of the cell cycle. And that's just to say that if you were to take a microscope and actually yourself watch it, you wouldn't be able to tell what's going on. You'd say, okay, the cell is not doing anything. However, the cell is actually doing a lot of stuff during this time. In fact, what's going on is the cell is increasing its supply of proteins and DNA precursor molecules, things like nucleotides. It's increasing the number of organelles it contains, specifically increasing mitochondria and ribosomes. And it's growing, it's getting bigger. And that's why I think a lot of people like to think of the G in the G1 phase to stand for growth instead of gap. So if that helps you and helps you remember what's going on during this gap phase, that it's growing, then that's great, and just use that. And so again, all that's going on in G1 is the cell's growing and it's producing organelles and molecules that it's going to use to then split off into two daughter cells. So what happens after the G1 phase? Well, the cell can do a few different things. It can move on to the next phase, which is going to be the S phase. It can go into this G0 phase, or it can be arrested in G1 phase. So let's consider each of these. So the first is pretty obvious, right? You just move on to the next S phase, and I'll talk about that in a sec. But let's talk about this G0 phase or this G0 phase. I was trying to remember if my physiology teacher taught it as calling that this G0 phase or the G0 phase. I can't remember. I think it's G0, but it just looks like a G with a subscript of 0. So if I say G0 and you're wondering what that is, it's the same as G0. And so this G, I'm just going to call it G0 from now on. So the G0 phase is considered the quiescent phase. And essentially what's going on is that cells can go into this G0 phase and stay in there for a semi-permanent or even permanent amount of time. In the case of a neuron, this is a permanent change. And that lets it essentially stop dividing and stop going through the cell cycle. So it's essentially like an exit lane to get out of the cell cycle. It's basically like that meme of that car that's skidding off the highway, off the side ramp where it's going and it's skidding into, that's G0. Going forward down the highway, that would be the cell cycle. So you can keep that in mind. G0 is just this phase that cells will sometimes take when they want to stop dividing. And importantly, this is a reversible phase. So cells can go back into the cell cycle if they want to. They can go out, then go back in, or they can stay there permanently, like in the case of a neuron. And I'm assuming that this is part of the reason why if you have a brain injury, you don't grow back that part of the brain because those cells can't divide, right? They're in this stable G0 phase. They're just kind of chilling, doing their job. Interesting. So that's one of the possible steps that the cell can take after this G1 phase. The other option is that the cell actually gets arrested in this G1 phase. And what do I mean by it gets arrested? Well, I'll talk more about this when I discuss the regulation of the cell cycle, but essentially the cell just kind of hangs out in this G1 phase and continues to keep growing and getting prepared to go into the S phase until it's ready to go into that S phase. And there's plenty of reasons why the cell might not be ready to go into that S phase. For example, and one of the kind of more important reasons a cell may stop and remain in that G1 phase is because there might be some kind of damage that's been done to the DNA that cell needs to go and repair, right? You don't want to have this mutated DNA that you then replicate and pass along to all the cells that are derived from this one cell. And that's how tumors are formed in some cases and how cancer grows. So it's important that the cell arrests, make sure everything is okay, and then it's like, okay, I'm good to go, and goes into that S phase. So let's just assume that this cell that we've been talking about decides that, okay, everything looks good. I'm going to move on to the S phase. So let's talk about the S phase now. So as I said earlier, the S in the S phase stands for synthesis, and that's because in this phase, DNA synthesis occurs, and specifically DNA replication occurs. And the cell needs to replicate its DNA in this step before it can undergo mitosis and actually split apart, right? Because if it doesn't do that, then you're not going to have two exact copies of genes that can be siphoned off to two different cells. You'll have one cell split off and have all the genes, another cell split off and have no genes if you don't replicate that DNA. And so during this phase, a few different things happen, right? You synthesize that DNA. The centrosomes are also duplicated. And for those of you who don't remember what a centrosome is just an organelle that essentially serves as like the microtubule hub of the cell. And specifically in cell division, it helps organize the microtubules that actually form the mitotic spindle, that's important for cell division. So that is another thing that happens during this S phase is the centrosomes are duplicated. And then protein production during this phase actually slows down quite a bit. And there is one exception to that, and that is that histone synthesis actually increases. And histones are just proteins that DNA is wound around that help kind of pack the DNA really tightly in order to fit it inside a cell. Because you always hear those stories of, if you unwound the DNA of one cell, you'd be able to reach the moon or something like that, or a basketball court or some length. They're pretty long. So I'm going to fact check myself real fast here. It's actually 2 meters long. So if you were to take a single cell and stretch out the DNA completely when it's unwound, it would be 2 meters, which is still a lot. Not quite to the moon, but that's still pretty big distance. And so these histones actually help the DNA get packed up so that it can fit inside these cells that are only about 100 microns in diameter. And more importantly, they actually play a pretty big role in gene expression. And I'm not going to talk about it here, but I think I'll talk about that in the genetics podcast. But they play a huge role in gene expression. All right, so to quickly summarize that, during the S phase of interphase, the DNA is replicated, the centrosomes are duplicated, and protein production slows way down except for the synthesis of histones. All right, so now the S phase has ended and we move into the G2 phase or the gap 2 phase. And again, you can think of this as the growth phase or the growth phase 2, whatever way you want to think about it, because again, the cell is undergoing a period of growth. And what happens during this time is the cell's growing, there's a really high level of protein production, and the cell starts to get ready to undergo mitosis. So some examples of the things that it does to get ready for this is that the cytoskeleton is dismantled to provide resources for this mitotic phase, and then the microtubules begin to reorganize to form the spindle. So again, during this G2 phase, all that's going on is the cell is growing and preparing to enter into the mitotic or M phase. And so the mitotic phase begins when the G2 phase ends, but of course, as was true with the G1 phase, there's other options that the cell can take before it enters that mitotic phase. And there's only one this time, and that option is that the cell can actually become arrested in this G2 phase. And I'll talk about this more when I'm talking about the regulation of the cell cycle, but essentially the same type of thing occurs where the cell makes sure that all of its DNA is replicated properly and all that's ready to go, and will kind of hold up that G2 phase, hold up the cell from entering into the mitotic phase from the G2 phase if it sees fit to do so. And let's just say that this cell has done so and it says, okay, we're all ready. Let's go into this M phase. So now you go from interphase, which is broken down into G1, S, and G2, and you enter into the M phase or the mitotic phase. And what happens during this phase is that the cell divides its copied DNA and cytoplasm to make 2 new cells. And as I'm sure you know, this process is called mitosis. And there's an acronym that helps me remember the different stages of mitosis, and that's PMAT, which is easy to remember because that's just the test that plumbers take actually to get into plumber school. That's typically how I remember that PMAT. And so PMAT just stands for prophase, metaphase, anaphase, and telophase. And I believe I talked about this a little bit in the genetics podcast, so I'm just going to play that audio for you real fast of what goes on during these different stages. Mitosis is somatic cell division, so essentially this is the division of your somatic cells, which are the cells that are not your gametes or are not your cells that can go on to produce babies. So let's start with the first stage, what happens in prophase. So in prophase, the nuclear envelope breaks down, chromatin condenses, the centrosomes separate and the mitotic spindle is formed. So you can picture this wild kind of flowy chromatin condenses into these X-shaped chromosomes. Your nuclear envelope starts to disintegrate, boom, and then your centrosomes separate, and again, your mitotic spindles are formed. And then the next stage, metaphase, kind of what it sounds like, the DNA or these chromosomes meet at the metaphase plate. And so I remember, metaphase, they meet at the metaphase plate. And this is due to the mitotic spindle pulling these sister chromatids via kinetochores. And so if you remember, kinetochores are these little center parts of the chromosome. So these mitotic spindles attach to the kinetochores, and they pull these sister chromatids to be able to line up at the metaphase plate. And then moving on to anaphase, once the chromosomes are all lined up, the centrosomes push out and the microtubules shorten, and this pulls the sister chromatids apart. So again, sister chromatids line up at the plate. Anaphase is pulling those sister chromatids apart. And then telophase, the last step of mitosis, basically everything that's done in prophase is redone. So the nuclear envelope forms, these chromosomes decondense, and the mitotic spindle breaks down. And then you'll have a simultaneous process called a cytokinesis, which is basically just the separation of two cells. So you form a cleavage furrow, and these two cells pop off from each other and produce two identical daughter cells. And it's important to mention that mitosis produces two equivalent cells. So you remember, these sister chromatids are just the exact same chromosomal information, just duplicated. So they're duplicates of each other. So when you pull them apart, yes, you have less total material, but you have still the same material is there. So it encodes all the same proteins, etc. You're not losing any information. You're losing the amount of overall information. All right, so that is mitosis, and that's what goes on during the M phase of the cell cycle. So the next thing I want to talk about here is a pretty important topic, and that is regulation of the cell cycle. And by the way, just listening to myself from that really old podcast is sad. I don't know, I sounded sad. I don't know what's going on that tape. I'm feeling better now.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
All right, so in this part of the podcast, I want to get into the regulation of the cell cycle. So according to a textbook called The Cell, A Molecular Approach, 2nd edition, the progression of cells through the division cycle, aka cell cycle, is regulated by extracellular signals from the environment, as well as by the internal signals that monitor and coordinate the various processes that take place during the different cell cycle phases. So in translation, the cell cycle is regulated by both external and internal signals. So that's the first thing to keep in mind. Both internal and external signals can regulate the cell cycle. And another important point is that there are 3 main checkpoints where the cell cycle is regulated at. The first being the G1S checkpoint, which is the barrier between G1 and the S phase. The second checkpoint is the G2M G2m checkpoint, which is the checkpoint between the G2 phase and the mitotic phase. And then the last checkpoint is called the spindle checkpoint. And this is a checkpoint that's located at the transition from metaphase to anaphase in mitosis. And essentially what a checkpoint is a point where the cell can kind of look at the contents of the cell, the state of the cell, and determine whether or not it wants to go over the threshold to this next phase. And so what I want to do is I want to talk about each of these checkpoints individually and talk about some important things that happen at each of these checkpoints and talk about a few different molecules that play important roles at each of these checkpoints, like protein P53 at the G1S checkpoint.
Checkpoint number 1, the G1S checkpoint.
So again, this checkpoint occurs literally at the gap between G1 and S. The cell can stay arrested in G1, as I was talking about, until it's ready to go into the S phase. And so what determines when a cell is ready to go into the S phase? Well, once the cell has adequately grown, it's grown to a good size, and has the proteins and adequate precursors / growth factors that it needs in order to replicate its DNA, and it's checked for DNA damage, that's kind of the big key here, then it can advance forward into the S phase. Because, you don't want to replicate that damaged DNA that could eventually lead to a tumor or cancer or something like that. And as I was saying a little bit earlier, the P53 protein actually plays a pretty important role here. It's able to halt the G1 phase when there is damaged DNA. And it does a whole lot more than just halting the G1 phase or arresting the G1 phase. It actually does things like promoting cell apoptosis, and a bunch of other things. But in terms of regulating the cell cycle, it's important to know that this P53 arrests the cell cycle at the G1S checkpoint. And because of its ability to actually arrest the cell cycle in G1, P53 is considered a tumor suppressor protein. And I'll get into more of this when I start talking about the application and diseases that are related to the cell cycle. But essentially, in short, mutations in the gene that encodes for the P53 protein can give rise to cancerous cells that can form tumors.
Checkpoint number 2. The G2m checkpoint.
The G2m checkpoint is at the end of G2 before the cell goes into the mitotic phase. And essentially what happens here is that the cell makes sure that the DNA is fully replicated and that there are no replication errors. And so being able to arrest the cell in G2 allows enough time for DNA repair enzymes to come in, fix the DNA where necessary, and then once everything's fixed and it's ready to go, boom, the cell goes into this mitotic phase. But really the two main things here are just you got to make sure the DNA is fully replicated. It'd be bad if you went into the mitotic phase without everything being fully replicated, and you check for DNA replication errors that may have occurred, because we all know sometimes DNA polymerase is known to slip up.
Checkpoint number 3, the spindle checkpoint.
So the spindle checkpoint, as I said, occurs during mitosis, specifically during the transition from metaphase to anaphase. And what this checkpoint does is it monitors the alignment of chromosomes on the mitotic spindle and actually ensures that a complete set of chromosomes is distributed accurately and correctly to each of the daughter cells. In other words, this checkpoint allows time for the chromosomes to meet up at the equator of the cell and then correctly get segregated to the two different daughter cells. Without this checkpoint, it's possible that you'd have a lot more cells that were missing chromosomes because, maybe it wasn't quite to the metaphase plate yet. A set of chromosomes weren't quite to the metaphase plate yet. And so they end up getting segregated together in a daughter cell. You might end up missing chromosomes because of this as well, which would obviously cause problems.
Cell cycle checkpoints complete. I have spoken.
So the last thing I want to mention in this segment is something that I mentioned at the very beginning, but you may be wondering about. So I said there are internal and external signals that regulate the cell cycle. So I want to give some examples of internal and external signals. And I wanted to mention the fact that molecules that regulate the cell cycle can act in one of two ways. They can either be positive regulators, and they tell the cell cycle to move on to the next phase, or they can be negative regulators, in which case they tell the cell cycle, hold up, stop where you're at, don't go on to the next phase. So just keep that in mind as I talk about these different molecules that regulate the cell cycle. In terms of internal regulators of the cell cycle, there's a few that are important. I talked about P53, which is a transcription factor. There's other transcription factors that control the cell cycle, but P53 is a negative regulator because, as I said, it tends to halt the G1 phase and not let it move on to the S phase. Another group of molecules that are regulators of the cell cycle are called cyclones and cyclin-dependent kinases, or CDKs. And these are proteins that are positive regulators and tend to move forward the cell cycle. In addition to that, another internal signal is cell size and also nutrient availability within the cell. External signals, on the other hand, are a bit more interesting to me, at least. So those are going to be like nutrients that are available to the cell from the external environment. Something like maybe cholesterol. Hormones are also another external signal that can regulate the cell cycle. For example, a group of researchers in 2002 reported in a paper that human growth hormone can actually stimulate cell cycle progression. In other words, it's a positive regulator. And to me, then I'd take that bit of information and I'd theorize that human growth hormone abuse could potentially produce cancer-causing cells and tumors because you're pushing these cells into further phases of the cell cycle before they're ready. And I have some anecdotal evidence that backs that up. I've had acquaintances, friends of parents that have been steroid abusers and then have actually gotten cancer later in life. And so that got me kind of interested just thinking about this. Is there a relation between HGH abuse and cancer? And so I'll kind of talk about this a little bit in the applications in disease part of this podcast. But really, the takeaway should be that you can have internal and external factors that can regulate the cell cycle, and those can be proteins, different types of nutrients, cell size, a bunch of different things. And these all kind of work together to push the cell into further phases of the cell cycle or hold it back at these various checkpoints. So in the last segment of this podcast, I want to talk about a relevant example of the cell cycle in disease. And the specific disease I want to talk about is cancer. And what I'm hoping this will do is I'm hoping that you can kind of tie some of this information to your knowledge on cancer already, and then maybe it might help you remember it a little bit better. Plus, it's interesting. Previously, I brought up this transcription factor in protein, which is known as P53, and it is a known tumor suppressor. There's a review paper that was put out by Chen in 2016 called The Cell Cycle Arrest and Apoptotic Functions of P53 in Tumor Initiation and Suppression. And what Chen said is that there are many roles of the P53 protein as a tumor suppressor that include the ability to induce cell cycle arrest, as I talked about, repair DNA, important for cell senescence and apoptosis, to name only a few. He goes on to say that the TP53, which is the gene that encodes this protein 53, or P53, mutations in this gene were reported to occur in almost every type of cancer at rates varying between 10% and close to 100%. In other words, a mutation in this TP53 gene is pretty common in cancer, which points at the fact that this protein, P53, plays a role in cancer. And so what happens if you have a mutation in this TP53 gene that results in loss of function of P53? Well, you no longer are as effective at inducing cell cycle arrest at the G1S checkpoint. And so then the cell cycle will move into the S phase from G1 without making sure that all of its DNA is okay. And therefore, mutations are going to start to accumulate over time as this cell continues to divide, continues to proliferate and go through this cell cycle, right? Because, maybe initially you have only a few mutations, but then it goes through the cell cycle again, and those cells then have more mutations that occur because the cell cycle isn't able to be kept in check by this p53 protein. And as these mutations continue to accumulate, you're eventually going to get a cell that has this cancerous phenotype where it is uncontrollably growing and dividing, proliferating and creating a tumor. The other thing I wanted to talk about was what I brought up earlier with the human growth hormone and my theory that maybe it can cause cancer by pushing the cell cycle through these checkpoints without it being fully ready to do so. And so I did a little research to try to see if there was any link between human growth hormone use and cancer, specifically human growth hormone abuse or steroid abuse in cancer. But I couldn't really find too much on that. I was looking at WebMD for a second, and it says that that may be the case, which tells us really nothing. But I did see that in a 2006 review paper that it indicated that there is epidemiological data that supports a link between growth hormone and cancer. However, they suggest that this is due to an increase in IGF, which is caused by HGH. And you don't have to know that. It's just a different pathway than what I suggested with the cell cycle arrest or alterations to the cell cycle arrest that may be caused by growth hormone. In terms of experimental studies, so epidemiological is not great for describing cause or for determining cause, right? It's great at finding correlations, not necessarily great at determining cause. So the next thing I did was I looked at experimental studies. And in mice, it's been shown that HGH or human growth hormone can increase the number of tumors compared to non-human growth hormone controls. However, there was another study that was an in vitro study, which is like in a test tube, where they took human gastric cancer cells and cultured them either in the presence of human growth hormone or not in the presence of human growth hormone. And they showed that essentially human growth hormone did not accelerate the cellular growth rate of these gastric cancer cells. So it's a little bit of conflicting results. Epidemiology suggests maybe. So I'm concluding then that there doesn't seem to be necessarily a consensus as to whether or not growth hormone or steroids can cause cancer. Doesn't really look like it. Kind of interesting to me. I think it'd be really interesting to take maybe a group of professional bodybuilders who have been 10 plus year anabolic steroid users, take them and compare the cancer rate in that group or cohort with a group of normal human beings that aren't steroid HGH users, and just see if the cancer rates are higher. I don't know if that's been done. That'd be interesting to see, in my opinion. And that concludes the Cell Cycle podcast. As always, thanks for listening. If you like the podcast, go give us a review on iTunes. Tell your other pre-med friends about it or other people who are studying for the MCAT. I hope this podcast has helped with your studying, and good luck to those of you who plan on taking the MCAT soon in the new year.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check out medschoolcoach.com for the experts.